So today what we're going to do is we're going to move on from the story of the neuron. I'll show you some pictures before we get into the glial story here in a moment. But basically yesterday was all about the structure of the neuron, the peculiarities of the neuron, and how we came to understand the organization, the functional and structural organization of neurons. And what we're going to do now, though, is we're going to move on into this new set of cells called the glia. And again, yesterday's lecture was all about the neurons, and most of the time when we think about the nervous system, we emphasize the role of the neurons, because the neurons are the electrically excitable cells. They're the ones that form the communication networks, and for the most part, for the last 120 years, we have emphasized on understanding neurons. Now, as time goes on, we're finding that the glial cells, the so-called glue inside the central nervous system and also in the peripheral nervous system, is playing an ever more important role. So I want to spend some time today talking about these glial cells, in part because I think within the next 10 or 15 years, we're going to find any number of neurological diseases, particularly the degenerative diseases, which we now ascribe to problems with the neurons, to be problems with the glia. Because what the glia do is they maintain an appropriate environment for neuronal function. They're basically keeping house. Doesn't seem like a very glamorous thing to do, perhaps, but this keeping house is really, really important. And when we look at what the glial cells do in the central nervous system, we see we can divide them into two big categories. The first category will be the astrocytes. And usually when we think about the astrocytes, we think about these guys as maintaining extracellular potassium and neurotransmitter concentrations. Now what we'll see is we'll briefly try to introduce the idea of a synapse here. We're going to drill down farther into that a little later. But when we talk about a synapse, one of the things you'll recognize in the central nervous system particularly is that there's not very much space. There's very tiny volume between cells. And there's also a very tiny volume between the output region of one neuron and the input region of a neighboring neuron. Because of these very narrow gaps, the synaptic clefts, the volumes are very small. Because the volumes are small, that you don't require very many molecules of neurotransmitter before you shift the concentrations by quite a lot. And so that's the role of the astrocytes. Make sure that these shifts in concentration for potassium or for neurotransmitter are not so large that they affect the function. So this is a regulatory role. The oligodendrocytes, and again, we're going to spend a little time on this, the oligodendrocytes are all about forming an ensheathing structure. Now, many of you may have heard of something called myelin. Myelin is a kind of insulation wrap around the axons. And it's the function of the oligodendrocytes to lay down these concentric rings of myelin. And we'll find that the concentric rings of myelin here are going to be basically the plasma membranes of the oligodendrocytes. And they run these concentric paths around an axon. And they grow around the axon to provide a kind of insulating layer. And we'll see a little later how this affects the electrical activities of the neuron. But for today, I just want to lay down the ensheathment. Now, one of the other issues here is that there's also, in the peripheral nervous system, a kind of glia. We give these a different name. We call these by this old eponymous name, the Schwann cell. But they're functionally glia in the peripheral nervous system. But the central and peripheral nervous systems have different functions, and they have different spaces. And what you find is where in the central nervous system, the spaces and volumes between cells are really, really small. Out here in the peripheral nervous system, they're much larger. And one of the other ideas I want to leave you with by the time we come to the end of the discussion is that the oligodendrocytes in the central nervous system form an ensheathment of the axons. That's true. But in addition, one of the things they do is they have proteins on their surface which inhibit regrowth of neurons. That's not true in the peripheral nervous system. In the peripheral nervous system, the Schwann cells, the glial and the peripheral nervous system, actually will permit neurons to regrow. They're actually helpful in regrowth. And this brings an interesting conflict. That is, in the peripheral nervous system, you have the glial cells that are supportive of neuronal regrowth. So if you damage your neurons in the periphery, there's some hope that they can regrow. That's not true in the central nervous system. In the central nervous system, damage to neurons leads to an inability to regrow, and this inability to regrow is largely thought to be because of proteins on the surface of the oligodendrocytes. This becomes a big problem, a big challenge in trying to sort out the mechanisms we're going to use to permit regrowth of damaged nervous systems. We all know someone who has had some sort of a terrible accident, and they've broken their spinal cords and that they're no longer able to control vast parts of their bodies. And we'd like to be able to fix that. But one of the impediments to repairing those damaged nervous systems is because these oligodendrocytes generate proteins which are inhibitory to growth. And we'll talk a little bit about that. Then if there's time, we'll come back to neurons, and we'll start talking about the electrical aspects of neurons. One of the hallmarks of neurons is they're electrically excitable. And as we implied yesterday, there are input regions, conductor regions, and output regions, and those differences in the regions were dependent upon the distribution of channels which are responsible for the electrical excitability of the neurons. Before we can really drill down into that, though, we really have to spend a lot of time thinking about how we set up the potassium battery. This is a fundamental idea in neurobiology, and the idea of the potassium battery is also applicable to muscle cells, and when we get to muscle, we'll talk about that as well. Now, this is a simple equation, and we'll simplify it even further. All it's telling us is that the equilibrium potential for potassium is proportional to the potassium concentration in the outside or the potassium concentration in the inside. This is the first step in describing how by separating potassium ions across a lipid bilayer, we can generate a kind of electrical gradient, a kind of battery. And then we can begin talking about how we can insert sodium channels to exploit that battery to send signals. And we'll spend a lot of time on that.